Welcome. We have introduced you to the structure of neurons, where we looked at the axon as the output side of neurons and the dendrites as the input side of neurons, and we briefly introduced to the idea of the structure called spines that are present on dendrites, which form the site of input to a neuron from a previous neuron. So, in this lecture, we will be talking about the structure of networks of neurons and the synapses that allow these networks to happen. So, we saw that Ramani Kahal's work showed that neurons are different entities, independent entities, and it is the synapses that make connections between different neurons, and this is where there is no continuity in terms of the inside of the previous neuron and the inside of the next neuron, and that is where the whole idea of the neuron doctrine is established. That is, through the synapses there is information transmission, but there is no physical connection between the insides of the two neurons. There are different other kinds of synapses, which are the electrical synapses, unlike the chemical synapses that we are talking about, which actually provide a direct connection between the inside of one neuron to the inside of another neuron, and that is a special kind of synapse, or those are special kinds of synapses, and they are present extensively during development and are present so that there is activity that is synchronized between neurons of different types, or different neurons can be made to have the same kind of activity, which may be required more so during development. But our discussions for this course will be mainly limited to the chemical synapses, which are the most important synapses because of their involvement in plasticity and information processing and so on. That is, the whole idea of learning is through synapses between neurons. So, let us go into sort of how we came to know about these things. Again, we refer back to work done by Ramani Kahal, and these are examples of their observations, or Ramani Kahal and some others' observations showing how intricate these connections are and networks are that form within the brain. So, both the examples here and here on the top two were created by Kahal based on observations. As you can see, that there are a number of fibers. This is, I believe, from the cerebellum, and there are a number of parallel fibers going in, which are making connections at different stages with the neurons here, whose dendrites are protruding up, and then their output is going through, where they are making again connections with other dendrites of other neurons. And similarly here we see that there are synapses that are formed on the neuron at multiple locations. So we had said that synapses form on what we call our dendritic spines, which are mushroom body like structures on the dendrite. As we said earlier, it is not necessary that synapses have to form on these dendrites, but they can also form, as here, on the cell soma itself, like we see here. So these are the axon terminals that are making connections onto the cell body, with making finger like projections onto the cell body, covering the entire soma, and these kind of synapses can exert very heavy control on the next neuron, like here. So these synapses are extremely strong. As you can see that there are many contact points. So the idea of the strength of a synapse will be coming up later on. For here, you can imagine it is almost like the many points of control are adding up to the strength of the synapse. So then the two other examples shown here with the arrow diagram show the flow of information as well. So there are a type of neuron that are providing information coming from the top, going down and providing an input onto one type of neuron at a next stage, and that axon projects on to the cell body of another neuron, and that proceeds, that sends the information out to the next stage. And this is how, again in this example also, this is how the system of neurons, that is the networks, are organized. So how the networks are formed during development, how the networks are changing or how they are labile, how they adapt to different situations, that we will take up in the last part of the course, is when we discuss about synaptic plasticity. So the basic idea here is that the input, so to speak, of a neuron comes onto another neuron through the structure called synapses. And a neuron can have, as you see in these diagrams, can have many different synaptic contacts from the same neuron, or can have multiple inputs from many different neurons, or they can also have simply one input. So the order of magnitude of the number of synapses that can form on a single neuron varies from 1 to tens of thousands. And so based on how these neurons are interconnected with each other through these synapses determine the function of the network. And we really do not completely understand how there are general principles that guide these connections to form. And although a lot of work has been undertaken to create what we call the connectome or the brain connectome, where we talk about exactly how the connections are made from neuron to neuron in different regions of the brain, those are open areas that people are working on. We do have some ideas of what is connected to what kind of neuron and so on, but those are those are basically scratching the surface. There is a lot of specificity of connections that form between neurons. So it's not that if this diagram says anything, it's not that everything is connected to everything. So there are only specific types of neurons that connect with specific types of neurons, and those are probably what need to be understood in order to fully understand structures of networks. So with this idea, let us go into a more deeper understanding of what we call the dendritic spines or the synapse with the presynaptic terminal and the postsynaptic terminal. So as you can see on the left, there is a entire jungle of neurons with dendrites and axons shown here, and from there are multiple locations at which synapses can be forming. So if we look at one particular neuron here, this is the cell soma, and all the dendrites are marked here with by marking a particular protein with fluorescence, and those allow us to see exactly where the dendritic spines have formed. So as you can see from this image, the cell soma uh, is dark, that is, on the cell soma there is barely any spine, although there are synapses on there. But as you go out on the dendrites, there are hundreds of spines which are the sites of formation of synapses with the previous neuron. So a blow up of a small region allows us to see here basically what a dendrite is like. So here is the dendrite going through, and on top of it there is a spine like so. That is, this part is the spine head, and this part is the spine neck. And as you can see, there are an immense number of spines that are formed. So each of those are sites of synapses. So what is actually happening in the synapse? What is the form of connection? How is information transmitted from one to the other? So that is depicted on the right hand side here. So as we said, if we consider the dendrite, say this is the inside of the dendrite of a neuron, and this is the neck of the spine, and this is the head of the spine. So this entire thing is the spine. Then a synapse forms on top of this, like so. This is the stereotypical structure. So this part is the axon terminal of the previous stage neuron. So if we have a neuron here whose axon is going, and then we have a neuron here whose dendrite is here with a spine at this location, if this neuron is making a synapse with it, this particular region is what is drawn here. So this side, the first neuron, neuron 1 here, and this is neuron 2 here, its axon is going out here. So this particular side on this neuron 1 side, this is called the presynaptic side, which is depicted here as the presynaptic side. And the spine side is called the postsynaptic side. Here the spine is not drawn completely, it is only showing this top part here, and this is the postsynaptic side. So in between there is a gap, and that is what we call the synaptic cleft. So how do we how is information transmitted from one to the other? We will be talking about it in greater detail when we discuss the synapses and model them computationally, but for now we need to understand the basic idea behind the synapse, and that is there is something in every synapse which is called a neurotransmitter. So these neurotransmitters are loaded in synaptic vesicles. So what the neurotransmitters are is that they are chemicals, they are molecules that act as a form of messenger from the presynaptic side to the postsynaptic side. What the neurotransmitter does, its mechanism of action on the postsynaptic side, that depends on the receptors of those neurotransmitters present in the postsynaptic side, and those are proteins that are expressed on the postsynaptic side. Okay, so we have these vesicles that are packed with neurotransmitters that are present at the axon terminal on the membrane in the just outside the synaptic cleft or just adjacent to the synaptic cleft. And when information needs to be transmitted from the presynaptic side to the postsynaptic side, that is from in this direction, it is the action potential or the membrane potential change that transfers from the soma and goes along the axon to the axon terminal and causes a membrane potential change in the presynaptic terminal. That allows a process by which the neurotransmitter gets released into the synaptic cleft here as described in this diagram. And then there are these postsynaptic receptors. They have sites where the neurotransmitter can go and bind and then cause changes or current injections into the postsynaptic nerve. So we will talk in greater detail about exactly how the action occurs, or rather how the next neuron is manipulated by the presynaptic side through these neurotransmitter and their receptors. We will talk in detail in later lectures. So, for now what we need to understand is that this synapse is the site of connection between two neurons. It is the information that is communicated from the presynaptic side that reaches the postsynaptic side via the neurotransmitter and the neurotransmitter receptors, the neurotransmitter being in the presynaptic terminal, the receptors being in the postsynaptic terminal, and then the way in which the neurotransmitter receptors behave when the neurotransmitter binds to the neurotransmitter receptor, that determines how the presynaptic neuron changes the postsynaptic neuron's activity or its membrane potential. So, the details of the manner in which these occur are well understood, and there are multiple different players in this little synapse, a few micron structure with nanometers level gap between them, and there are intricate details that actually control different behaviors at synapses, and how these synapses change over time will be probably the most important part of the course, in the sense that allows us to learn how processing occurs, how learning occurs, how memory forms, how we adapt to different situations, how we make decisions, how we change at fast time scales, and so on. All these things are dependent on these particular structures, heavily dependent on these particular structures called synapses. So, as I was mentioning, it is not necessary that the synapses have to form on spines like here, like we have described here and here. The synapses can form directly onto the dendrites. So, if this is the dendritic shaft, then a synapse can form like in this manner on the dendrite itself. So, this is the presynaptic side and this is the dendrite of the postsynaptic side. So, there is no spine here. Even the spines can be of different forms. They can be the narrow neck and mushroom like structure, they can be thin filipodia like structure, and so on. Similarly, the synapses can also form directly onto the soma, as we saw in the previous slide, where there are number of synaptic contacts made by the same neuron on the soma of that neuron, of another neuron. So, now the most important thing that we have not discussed and just alluded to is the currency of computation or the action potential, not just the computation, but also communication between neurons. As we said, that information in some form is coming to the synaptic terminal or the presynaptic terminal, and that is being communicated to the next stage through the synapse, that is being communicated from the presynaptic side to the postsynaptic side through these receptors via the neurotransmitters. So, what is it exactly? What is it that carries this information? And these are what we call spikes or action potentials. So, we will discuss in great detail when we understand how this action potential occurs, and for that we will be looking into the revolutionary work of Hodgkin and Huxley, who also got the Nobel Prize in Physiology and Medicine in 1963 for their work in the late 40s, early 50s about understanding quantitatively how the membranes of neurons can produce action potentials and how it is transmitted from one neuron from the neuron along its axon, and how later on how that influences synapses and information is transmitted from the previous structure to the next structure or previous neuron to the next neuron. So, what is this action potential? So, if we were to measure or record somehow, as described here, the membrane potential of the neuron from its soma, that is, as we had drawn Let us say this is the membrane of the neuron. We have a lipid bilayer as described here, which separates the inside and outside of the neuron. Let us say this is the inside and this is the outside. There are different ions at different concentrations on the two sides of the membrane, and that creates a difference in the potential in the two sides. So, if the potential inside is V in or V I, potential outside is V O, then this difference V I minus V O, which we will refer to as V membrane or the membrane potential or delta V sometimes, or even sometimes just V. We will, depending on the different cases, we will be using all these terms, and this is the basis of all the computation, this membrane potential. And if we were to record, as depicted here, the membrane potential of a neuron, so that is shown here, then this along the voltage axis, there is a point at which the neuron stays, the membrane potential stays at rest. That is, when there is no activity, when it is simply stationary and there is no perturbations onto the neuron, then the membrane potential stays constant based on the equilibrium of the different ions, and that turns out to be around minus 60 to minus 70 millivolts, around that. It varies from neuron to neuron and during different states also. And gradually, because of this input from the previous side and its influence on the post side, because of the neurotransmitter, there may be changes in the membrane potential. That is, it can be excitatory, where the membrane potential increases, or which we call depolarization, or it can be inhibitory, where the membrane potential decreases, which is what we call hyperpolarization, bringing the membrane potential down towards the negative side. So, in this, implicitly we have already introduced two different kinds of synapses. Synapses that are excitatory, that is, they move the postsynaptic membrane potential upwards. Synapses that are inhibitory, that push the membrane potential downwards or towards the resting state. So, depending on the kind of inputs coming onto the neuron, as we saw, there can be thousands of inputs coming onto a neuron, and what influence it has on the membrane potential of the neuron, that can be recorded by recording this membrane potential. And as you can see, the membrane potential changes, and there is a point which we call a threshold potential, or VT. When the membrane crosses that potential, there is an event called the action potential that shoots up the membrane potential up to plus 20 millivolts or plus 40 millivolts, around there. So, this is a huge excursion from minus 60 to plus 40, around 100 millivolt excursion within a very short time period. This is a millisecond or so, a few milliseconds, 1, 2, 3, depending on the type of neuron, but it is indeed very short and very fast. This event, which we will show later on, is all or none, that is, it is either occurring or not occurring, and it occurs whenever it crosses the threshold. This event is the action potential or spike, and is the currency of computation and communication between neurons. So, this action potential or this change in membrane potential, when it travels from the soma along the axon to the axon terminal here as described, then there is a process by which this neurotransmitter gets released. So, that is where the connection of the communication between neurons and the computation by the previous neuron is coming in. It is the action potential that is generated based on computation in the soma or the axon hillock, as we described in the very beginning of the axon, and that is communicated via the synaptic terminal to the postsynaptic neuron to produce an excitation or increase in membrane potential on the next side, or inhibition, that is decrease in membrane potential on the next neuron through these synapses and the neurotransmitter and its receptors. So, this completes the full circle. Now, this membrane potential change in the postsynaptic neuron, whether it is excitatory or inhibitory, and how other inputs are coming in over time, that determines how the processing or computation done by the next neuron, the postsynaptic neuron, is determined. And if it produces a series of action potentials, then those action potentials are again transmitted or communicated along that neuron's action terminal to the next stage neuron, and so on. So, this is sort of the basic crux of how the information is flowing in the nervous system, or in at least in the central nervous system, which we will try to understand in terms of how these action potentials are generated quantitatively. We will also understand how the series of action potentials produced by a neuron actually carry information about the external world or the mental state or what have you. And in the last part of the course, we will be talking about how these synaptic structures are modulated, or rather how synaptic plasticity occurs based on the activity of the presynaptic side and the postsynaptic side in a self-organizing manner. That is totally activity dependent and self-organizing. That is, based on the activity, the synapse itself modulates or changes itself and the system learns. So, with this we come to the end of the structure of networks of neurons and networks of neurons and synapses lecture, and we will carry on in the next lecture with information flow in the nervous system. Thank you.